Hello everyone, this is Bill Errickson. I'm a PhD student at Rutgers University working with Dr. Bingru Huang, and this project highlights some of our current research using plant growth promoting rhizobacteria to improve drought tolerance and post-drought recovery in cool season turfgrass. This research is supported by a novel Approaches to Research grant from the USDA Sustainable Agriculture Research and Education Program. Cool season turfgrass, such as creeping bentgrass, can be severely affected by drought stress, resulting in leaf senescence, a thinning of the turf canopy, and a reduction in turf quality. These symptoms are caused by an increase in the hormone ethylene in response to drought stress. Plant growth promoting rhizobacteria are microorganisms that can form a relationship with the roots of plants. Some rhizobacteria can produce an enzyme called ACC deaminase, which effectively reduces ethylene levels in plants, thus limiting the impacts of abiotic stress. Our team at Rutgers University has discovered several novel strains of rhizobacteria that were isolated from the roots of native grasses and exhibited high levels of ACC deaminase activity. Our previous work with these novel plant growth promoting rhizobacteria has demonstrated improved drought tolerance of cool season turfgrass in controlled environment growth chambers. However, effective bacterial inoculation in field conditions is more challenging due to the presence of native soil organisms and fluctuating environmental conditions. The objectives of this project were to determine whether inoculation of roots with ACC deaminase bacteria can improve drought tolerance and post-drought recovery for creeping bentgrass in field conditions, and to evaluate phenotypic traits of creeping bentgrass regulated by ACC deaminase bacteria in association with drought tolerance and rapid recovery upon rewatering. This experiment used two novel strains of rhizobacteria combined with humic acid to inoculate the roots of creeping bentgrass maintained at fairway height in field soil conditions at the Rutgers University Horticultural Research Farm 2. The rhizobacteria inoculant was applied as a soil drench and compared to non-inoculated controls and a commercially available bacterial inoculant. The plots were inoculated with a soil drench method twice before the initiation of drought stress and once again before the rewatering period. A rainout shelter was used to exclude rainfall from the plots. Deficit irrigation treatments were applied weekly at a rate of 60% ET for 49 days. After the deficit irrigation period, the plots were rewatered to field capacity and full irrigation was resumed during a 28-day recovery period. Weekly measurements included turf visual quality, rated on the basis of color, uniformity, and density on a scale of 1 to 9, and normalized difference vegetation index, or NDVI, values were measured with a crop scan multispectral radiometer. We also used a light box to capture uniform digital images of the plots throughout the growing season. These images were analyzed using the SigmaScan software program to quantify percent green cover and dark green color index of each plot. Plants inoculated with the novel Rutgers PGPR strains demonstrated enhanced drought stress and post-drought recovery. Lightbox images taken from the same location each week depict the progression of drought stress and recovery in creeping bentgrass inoculated with the growth-promoting bacteria. Inoculated plots retained higher turf quality and percent green cover throughout the drought stress period and during post-drought recovery. NDVI and DGCI also remained higher in the inoculated plots. These results suggest that our novel combination of Rhizobacteria strains effectively improved drought tolerance and post-drought recovery upon rewatering for fairway height creeping bentgrass in field conditions. As such, these novel plant growth promoting bacteria strains have potential for development as a biofertilizer for reducing water use in cool season turfgrass. ACC deaminase activity of the bacteria was the likely mechanism for improving turf quality during drought stress, though further research into the mechanisms involved is needed and will be the focus of future research. Thank you for your attention, and please feel free to contact us for more information.